position of the molecule inside the nanochannel. So this trick is a core of our technique that enables us to see invisible things, such as biomolecules. So what you can see here is a real-time video of a DNA molecule. Specifically, it's a lambda DNA molecule, which flows through a nanochannel. It flows through the nanochannel because we introduced there a pressure drop. So it's basically pushed from one end to another. So this molecule is actually quite big. It's like 30 megadaltons molecule. It's actually our first experiment, and we were quite happy what we saw. But then we started to push the limits of our method, and we started to see smaller and smaller things. Like, for instance, this is a video of a ferritin molecule. It's a molecule that has 400 kilodaltons, and this molecule, as you can see, like freely moves inside a nanochannel. The movement you see is basically a Brownian motion of the molecule. So in this example, we don't push the molecule from one end to another. We just basically let it diffuse. And this, what you see here, it's a library we have succeeded to image over time. All these images are basically like a time-lapse images, so-called chemographs, where every line corresponds to one time frame, basically averaged over the short axis of the nanochannel. So these lines, what you can see here, corresponds to basically a trajectory of single molecule. And you can also see here that as the molecule becomes smaller and smaller, these trajectories become more wiggly, because the Brownian or the diffusivity basically increases when the molecule gets smaller and smaller. So the Brownian motion becomes faster and faster. So we succeeded to image molecules ranging from tens of megadaltons to molecules that are, right now we also can see like smaller molecules than this 100 kilodalton DNA. We can go down like to 100 kilodaltons, let's say. So to make this happen, we use an optical microscope. In principle, it's basically a dark field microscope, which outperforms the conventional microscopes in terms of mechanical stability. The method requires a sufficiently powerful source of light. In our case, we use supercontinuum laser, and also requires a high-speed camera. In our case, we use a CMOS camera from Andor. And an essential part of this method is a fluidic chip that contains a series of nanochannels that are connected to these sample containers through these like two microchannels. And the flow of the molecules through the nanochannels is controlled by pressurizing the inlets. But we cannot only see the molecules, but we can actually extract a lot of useful information from the image. For instance, from the contrast of the spot, we can determine polarizability of a molecule, which is closely related to a molecular weight. Basically, it's a very similar concept that was introduced by IceCut, for those who know this method, where they use basically the same principle to weight the molecules using light. But apart from the information about the molecule mass, we can also determine the diffusivity of the molecule from the statistics of its movement, and from there extract information about the size or conformation of the biomolecule, which is related to the hydrodynamic radius. So from those images, we can extract two sets of information. Information about the mass and information about its size. So here is a collection of measurement of different molecules, like 3 short DNAs and 2 proteins. Each point in a graph corresponds to a combination of values of optical contrast and diffusivity pertinent to every single molecule that was detected. For every type of a molecule, we can see a distinct population with optical contrast that linearly increasing with molecular weight. But we can also see clear difference between the diffusivities of DNAs and the proteins, resulting from the fact that the diffusivity depends not only on the size, but also on the shape or conformation of the molecule. That is very different for elongated DNAs and globular proteins. In addition, for proteins, you can see, like apart from this main peak, you can also see a different peaks that corresponds to dimers and higher oligomers, maybe that we cannot see here in this picture, but also like for instance, for this ferritin molecule, you can also see some additional peaks that at this point we don't know what they are. They might be perhaps dimers with weaker interaction, or there might be different population of molecule that contains maybe different amount of metal ions. Anyway, if there is anybody in the audience who might help us to clarify it, it would be great. Yeah, apart from characterizing the mass and size of a molecule, we believe that this method could also be used, for instance, for investigation of biomolecular interactions. On this example, I show a captured event of creation of a complex. At the beginning, we see 2 molecules of ferritin approaching each other and creating a dimer. Or in the second case, we see the interaction of the molecule with the surface. So at the beginning, we see freely moving molecule that at some point binds to the surface and unbinds, and then continues freely diffusing. At the end, I'd like to add that very recently we have founded a spin-off company called NView in a collaboration with Chalmers Ventures and Chalmers School of Entrepreneurship. And at this early stage, the company is mainly looking for incubation opportunities, but also for partnership within the scientific community to help us explore the full potential of our method for applications in biochemistry, biotechnology, or drug discovery. With that, I'd like to summarize. I have introduced a microscopic method that allows for label-free imaging of freely moving single biomolecules of sizes down to 100 kilodaltons in molecular weight. This method also allows us to measure the molecular weight and size at the single molecule level. And at this moment, we are really at the beginning, but we hope that this method will find its applications in single molecule mass spectrometry, investigation of biomolecular interactions, or particle counting. But it would be very interesting to hear from you your immediate ideas where you can see the potential of our technique or how it can be used for your applications. At the end, I'd like to acknowledge my colleagues from Chalmers, from different departments. And I'd like to thank you for your attention.
Barbara, thank you very much.
Okay, indeed, yeah, I should leave the word probably to Tatiana, who runs the show. I expected that maybe I should then moderate the discussion, but if Tatiana can do it, I would be happy to let her do it. Sorry for stepping in.
Yeah, thank you very much. Thank you very much, Bara, for a very nice talk. It's really interesting and innovative technique. I have also my personal questions. First of all, who wants to ask the questions, please raise the hand or just write your question in the chat. So now I can see only one raised hand. So I have my personal question. What are the limitations of this method? Because from the poster, I know that up to 100 kilodalton, like let's say megadalton particles you can observe.
Yeah.
What, for example, is the lowest?
Well, the lowest what we could see now is perhaps like a BSA molecule that is like 60 kilodaltons, but that's really at the edge. Yeah, that's the lowest limit in terms of size.
Mm-hmm. Do you have some limitations, for example, for the buffer composition or, for example, salt, if it should be, I mean, of course, because every method is really sensitive for the presence of some ions or, for example, some bigger organic solvents. So what are the requirements?
So basically, like in terms of like optical performance, there's no limitation in terms of ions inside or different buffers. But right now we use just bare nanochannels, so we don't use any coatings. So just for these proof of principle experiments, we just selected biomolecules that have relatively low pI, so they don't actually absorb on this surface so much. You know, when some molecules that would absorb more on the surface, it would be kind of difficult to analyze them because they would be absorbing and desorbing in like more frequently. But we plan to use some coatings, like some that would prevent basically the molecules to absorb. Mm-hmm. That's our current limitation, maybe.
Mm-hmm. And how much, because it's nanotechnology, and of course you have just this microfluidic technique, and how much actually you need the protein, I mean the amount of the sample?
Yeah, so usually, you know, this chip is mounted to our sample holder. So basically what we need is like a drop. We usually use like 10 microliters. That's our volumes that we use. So it's really tiny.
Yeah, and at which concentration, for example?
And concentration. So basically, what's like best or what we work with is basically like 10 nanomolar kind of concentration, because that concentration basically results in like one molecule in a field of view. So we, of course, go to much lower concentration, then you would have to be just patient and wait long enough to basically find some molecule. But we probably cannot go much higher with the concentration, because then that would result in many molecules inside one diffraction limited spot, and that we would not be able to discriminate basically. So it's like a nanomolar concentration kind of.
Okay, perfect. Thank you very much for the answering my questions, because it's really interesting technique, and I think it's really very useful. And after your presentation, I wanted to try also my samples. So hopefully I will have also the chance very soon to apply this method. Thank you very much. I will ask another guests for their questions. Okay, Jakub, can you ask your question.
Yes, hello, Bara. I'm wondering, I mean, thinking of how to extend this technique for the interaction studies. I mean, would there be chance to somewhat introduce to your channels, for instance, some, I don't know, lipid bilayer system, or, I mean, is that the plan to kind of, let's say, mimic some, let's say, biological environment in these channels and then observe the interactions there? And would it make sense to observe the interactions between somehow tethered ligands and then your affinity partner, for instance, freely diffusing through the channel, or should both the interaction partners be diffusing in the channel, like the example you showed?
Yeah, I think like in principle, we could do both. You know, we could observe the interaction of a freely moving molecule with some molecule on a surface, but that's, you know, what IceCat, for instance, can do. So so what we feel and what's like the biggest strength is that we could see both molecules just freely diffusing. So my personal opinion is that it would be just super cool and super nice just to see like two freely moving molecules interacting, you know, because that could avoid like some biases that are introduced in like more conventional interaction studies where like one molecule is attached to the surface and then you observe interaction with a molecule that is already kind of modified because it's attached on the surface.
Yeah, but I mean, lots of biological systems, yeah, like I don't know, protein interactions in lipid membranes, they require this kind of environment. And I think your, let's say, channels are too narrow to load in some lipid vesicles or stuff like that. Or yeah, but yeah, maybe you can make them wider.
I mean, like coating of nanochannels with lipid bilayers is like a standard thing that our colleagues in Chalmers are actually doing for different purposes. Basically like studies of DNAs. The lipid bilayer is something we could push inside. But then, you know, it's a question also how you actually modify the interaction, because in our case the molecules are kind of squeezed in very confined volumes. So I don't know if that could be, for instance, interesting for studies in I don't know, to mimic some biological systems where you have like long nanochannels or I don't know, something like this.
Yeah, but I guess that's what the biology folks will come to. Thank you, Bara. Thank you very much.
I'm inviting also Adam Bolja to ask his question.
Okay, hello everybody. And my question is, can you use your method to somehow actively interact with the molecule? I mean, for instance, by changing the wavelength of the light. I mean, some molecules are sensitive, for instance, for the ultraviolet. So you can like use it to change the molecule in the channel. Is it possible or not?
Like boil it, basically.
Yes, for instance, or for instance, divide it on two parts. For instance, just applying some high intensity photons on some specific wavelength, if it is possible.
Well, yeah, I mean, in principle, you know, if that is something that would be desirable for some applications, that then you can introduce some probably like different light beam with different wavelength. Actually, our super continuum starts at I think like 350 nanometers, and ends at 2 microns. So we have quite wide span of wavelengths that we could use. But yeah, we have been, for instance, because when I started to work with this technique, I started with basically DNAs that were labeled by some fluorescent dye. And what we have been doing is that when I used the excitation wavelength of the yo-yo, you could basically observe the how the DNA actually breaks into several parts.
Okay, yeah.
So, I mean, if you would like to see, I don't know, breaking of some molecule into 2 parts, you can maybe use like some label and then use the light for it, for instance. I don't know, like, do you have something specific on your mind, or?
No, I don't have anything specific, just seems to me very interesting, so this is a question that I also wanted to ask if you can actively move somehow the molecule, but I think it's too tiny. It's definitely.
No, like you mean like optical tweezers, or?
Yes.
Yeah, that's too tiny.
Okay. Yeah.
But we can play with the flow a bit. Basically, move the molecule back and forth.
And some apply some external electric field.
Yeah, you can. Yeah, like for instance, how the electrophoresis is done. It's probably that. But for now, we're working with basically like pressurizing the inlets. It's how we flow molecules. But yeah, you can also think about, I don't know, introducing some kind of like electrostatic traps. You can trap the molecule and maybe observe it for a longer time if that's needed. But that's all, yeah, just ideas right now.
Okay, yeah. Okay, thank you. Thank you for answers.
Thank you very much. I have also a question probably about the dye. Should we use special type
to label our sample? I'm sorry, could you repeat the question?
Yeah. Can we use any type of dye to label our sample? Or do we need special type?
Well, so this technique actually is like it works also without any labels. So, but if that's needed or desirable, we can also work with labeled proteins, and actually we can do both at the same time. We can basically do the like fluorescence imaging, but also we can do this scattering imaging at the same time. It's something I haven't shown, but that's also possible. Yeah.
Do we have some more questions? Who wants to ask something? In this case, I would like to thank Bara one more time. Thank you very much for a very nice talk, very interesting answers. And I think we can continue with our second speaker.
Okay, thank you.
Thank you very much. So, Dr. Eugene, are you here?
Yes, I'm here. Can you see me?
Yes.
Can you hear me well?
Yes, yes.
Okay.
Can I ask you also, if I'm pronouncing correctly your name, so Eugene O?
Yes, perfect.
Okay, thank you very much. So I'm welcome you, and I will introduce the Dr. Eugene O. She is from the Institute of Biophysics, Johannes Kepler University from Linz. And now she will tell us bit more about the force spectroscopy and recognition imaging. She will tell us about the quantifying binding strengths and affinity in the single molecule level. So we will continue again with single molecule level and single cells for spectroscopy. And she will tell us about more the dynamic and the statistical information about this technique. This nanomechanical actually behavior, and I'm really welcome to hear more about this.
Hello. Yeah, can you see also the screen now?
Yes.
So also presentation mode. Yes. Okay, then yeah, good afternoon, everybody. Yeah, first of all, I also thanks to organizer to give me the opportunity to share my research and also also my recent publication. So yes, as you pronounce it correctly, my name is Eugene Oh, and I'm currently working in the Biophysics Institute, Kepler University, Linz. And today I entitle my talk the force spectroscopy and then recognition imaging. Then I will show you about how we use basically these AFMs, the scanning probe microscope or atomic force microscope in our group. So relatively kind of length introduction. And I also want to bring our recent publication, how we use this AFM to quantify the binding strength and affinity of on the DNA sensor. So just wait, data pointer, yes. And I mean, as we are all working in the nanotechnology, probably here everybody knows about the scanning probe microscope or the atomic force microscope, which was invented kind of 40 years ago. And just to refresh up how this AFM work is we use this AFM cantilever here and basically scan the surface and we detect the movement of our AFM cantilever using the laser and then detect with a 4-segment photodiode, and then we can convert basically the 3D structure of our sample surface. So here, as you can see, the schematic diagram of the AFM, and it has quite a bit advantage. It's not only the high resolution of AFM like ideally in vacuum condition that resolution, but it can be also operated in the different various environment. For example, like a liquid. And as we are interested in the biomolecule or cell or bacteria and so on, like AFM can be operated in the physiological condition. And it can also apply, we have kind of quite a bit the application way. So we can also measure the current or the magnetic force or electrostatic force and so on. So AFM is now a very common technique in the nanotechnology. And I want to bring first example how we use this AFM for imaging of the biomolecule. So I brought the example sample as a bacteria because bacteria is kind of size-wise is 1 to micrometer size. So it has quite a bit difficulty to see the single bacteria with a common optical microscope. But with AFM, we can see better. But meantime, bacteria is pretty mobile in liquid, so they can swim around. So it's also not easy sample to measure with other microscopic technique. And here is the bacteria sample. So this is the Tannarella forsythia, which is the oral pathogen, famous kind of red complex family, and it causes the periodontal disease. And it is known as it has kind of surface protein structure on the bacteria surface to kind of promote or help for the better adhesion in our tooth or teeth environment, in the oral environment. So first, when we apply the most known contact mode, AFM imaging, as you can see, the basically cantilever scan the surface and then we detect the movement. And then we can see quite a bit detail of this single bacteria surface. But the problem of this contact mode AFM is it often damages the soft surface, like a cell surface is very soft, and then as we scratch directly on the surface, it damages the surface. So the other imaging method is so-called the tapping mode or non-contact mode. So we have cantilever oscillating in the resonance frequency, so fixed frequency, and we detect the change of amplitude due to the van der Waals interaction between the tip and the surface. And this is quite good to measure the soft sample, especially for the biological sample. And then when we measure this bacteria surface in the physiological condition, we using tapping mode. And then we could clearly see the protein lattice structure, as you see here. And then when we Fourier transform this image, and then this lattice structure of the surface protein layer on living bacteria has about the 10 nanometer lattice, which is pretty fit well with the previous electron microscope result. And also angle is pretty comparable to the TM measurement. So you can see really AFM shows the very high resolution in the living organism in the physiological condition. And it can also apply not only the very small protein size, also the bigger size like the macro scale, like a micro scale, which means like a 10 to 100 micrometer. And here is the example how these bacteria form the biofilm on the abiotic surface. So this E. coli, basically we grow the biofilm of E. coli on the glass surface. And then as you see, as time goes by, the first they form this small microcolony, just a few bacteria adhere to the surface, and then they form really from biofilm. And after a certain time, they also detach it to basically to expand their biofilm amount in the other place of the surface. So that's kind of typical behavior how biofilm form. And some of bacteria like E. coli or Salmonella, they produce also other adhesive protein, which called like a pili or coli. And here you see also flagella of the bacteria. And then when they have this kind of coli adhesive protein, more, then they also start to form more amount of biofilm, which we could clearly see then. And also 72 hours, they completely cover all the surface and really produce also extracellular polymeric substance like to really make the mature biofilm structure. So I would want to say that AFM is really great tool to show the image, not only the single molecule level and also the macro size like a bacteria or single cell surface. And another advantage of our AFM is we can use as a force tool. So here it's so-called the force spectroscopy mode. And we can move AFM cantilever to the surface up and down. And then these laser deflection traces will go like this, as you can see, go up, and then as it cantilever bends, and then also go down. And when they detach, then they go normal position. And we can apply this for spectroscopy technique also to the biological sample. So as you can see, the biological samples are a bit softer than the harder surface. And then you see this kind of soft indentation here. And then furthermore, they finally detach it, showing the specific interaction between the receptor on the membrane and then ligand, what we are interested in, the AFM cantilever. So we can modify our AFM AFM cantilever with really different ligand. We have quite a bit library to attach whatever ligand on our AFM cantilever. And most of AFM cantilever made produced by the silicon-based MEMS technology. So we can apply the easily the amine group to the end of AFM cantilever tip, and then covalently link the polyethylene glycol linker to the end of the tip. And here, end of the spacer, we can linking any ligand here, for example, antibody or other single protein or peptide. And why we give this kind of PEG spacer, which is about 5 to 6 nanometer length, is because of it can also make this ligand freely move and then orient it rapidly when it interact with the receptor on the membrane or on the surface. And this force distance cycle give basically the force what we measure. So as you see, this unbinding force, interaction force, so-called the dissociation force between the ligand and receptor complex. And we can also measure how often it happens, so like a binding probability. And further, we can also calculate their energy landscape of this molecular complex. This is also an example of our single molecular force spectroscopy to the sample previously I mentioned. So as I said, the E. coli or Salmonella produce like this Colli fiber structure for their attachment. We also confirm it in the abiotic surface, but we are also interested that they really also work. They produce it when they bind to biotic surface. So how we perform the experiment is like we prepare this Colli fiber monomer on the surface. And then we modified our AFM cantilever with the 3 different types of the protein. So first is the fibronectin, which is the extracellular matrix of the tissue part. And we also kind of cut off this fibronectin domain 3 part here. And even we can cut off this one domain from this fragment, so RGD peptide. And we basically link covalently with the 3 different way. One with the lysine residue, one with the histec protein, and one with the cysteine residue. We can covalently link them and then measure the interaction. So when we see their force traces, and then as you see, we see this specific interaction between our Colli monomer and then fibronectin fragment. And when we perform this experiment many times, like 1,000 to the 5,000 or 10,000, we can collect enough statistic to carry out what is the really highest or most common this unbinding force is. So you can see in this the distribution of force, and then the probability density function shows basically how often it shows it. And as we can see, all 3 fragments shows the kind of similar regime force, which is about the 50 piconewton, which means in the fibronectin part, the RGD domain is the peptide is the main part of the interaction, even though it's really very small in this long, the dimeric extracellular matrix part. And furthermore, we can also vary the pulling speed of this experiment, so change the pulling speed, and then plot it with the force versus loading rate, and apply the simple single energy well model, which is the Bell-Evans model. And we can carry out basically the kinetic off rate of this interaction. So from the slope is the kinetic off rate, and also X beta, which is the distance between the energy minimum here, the bound state, to the unbound state. And in this case, all 3 molecules shows the similar kinetic off rate, which means the RGD is the main interaction. But meantime, the X beta is varying a little bit, some intense due to the length of the molecule or the structure of the molecule is different, and then this flexibility of the binding pocket change due to the, basically the size of the molecule. And furthermore, we can also apply this single molecular force spectroscopy, not only the isolated protein, also on the living bacteria surface. So we tried it 3 different mutants, like a wild type, and then this Coli knockout mutant and Coli overexpressed mutant. And all 3 partner shows really pretty much low, no binding effect, which means the Coli is really interacting with the RGD of the fibronectin. And then Coli overexpressed mutant shows the much higher, yes. And when we extend our chemistry a little bit, and then we can also directly attach the single bacteria to the AFM cantilever like this, and also measure the single cell force interaction with the surface. So in this case, we coated the surface with the fibronectin and then measure the one E. coli cell on the surface, how they interact with. And as we calculate the force and distance here, that we can easily calculate the work, how much required to detach one single cell from the surface. And as you can see, that this overexpressed mutant require much higher work to detach for the detachment of single cell from the surface compared to the wild type and the knockout mutant. So we play a little bit calculation here. So basically, we can also calculate the contact area between the single cell and surface using the Hertz model. And then we can estimate the kind of how many molecular bonds form in this case. So in one bacteria can have 9 to 10 bonds formation with the fibronectin surface. And as we calculate the kinetic off rate from the previous fitting in the single energy well, the model, we can calculate also lifetime of our molecular bond, which is about the 0.8 second. And then when we apply the 9 to 10 bonds, and then it finally lead us kind of the 3 seconds simultaneously unbinding, which means the all molecular bonds disrupt simultaneously, or the sequential behavior like a one by one detachment. In this case, the lifetime of our molecular bonds increased dramatically from 2 to 8 seconds. But still, nevertheless, 8 seconds is a bit short. I thought that when one bacteria binds in my intestinal cell, and then how long they stay on the surface. But this is the calculation according to when there are single protein on the surface distributed on the bacteria membrane. But as you can see, the bacteria form not only the single protein on the surface, they make this kind of fiber structure, which means that they can always rebind and rebind to the other molecule while one is interacting to the other. And when we consider this kind of long-lasting interaction or the rebinding effect, then this the lifetime of bacteria at the molecular bonds will increase even furthermore, which shows why bacteria basically produce like a fiber structure, not the single protein structure. So that was one of example how we apply also for spectroscopy. And with this, as we can now attach the single cell to the AFM tip, we can directly measure the single cell interaction with a different abiotic surface. So in this case, we got the basically this catheter sample, which is used for the stent in the body. And they coated this catheter surface with a polyethylamine polymer brush with a different molecular weight and a different arculate. And when we see also, when we measure the one single bacteria interaction with this catheter surface with a different polymer brush grafted, And we see this higher molecular weight of the polyethylenediamine polymer brush with the arculate shows really no forces, which means that they completely repel the interaction with the bacteria. And we can also apply to different antimicrobial polymer brush surface, like a cationic or anionic and neutral surfaces. And as E. coli is negatively charged, and when they interacted with a different charged surface, they show pretty completely different binding behavior. So like a glass or the cationic surface, they binds well. But when they are the same charged surface, they start to repel, and then that shows antimicrobial effect of the surfaces. So I can say that now we can apply really many different surfaces to test the antimicrobial effect or more the biocompatible effect between the cell and the surfaces using our single molecule force spectroscopy technique. But now I would like to actually use this AFM not only for imaging or to figure out the interaction with the other biomolecule. We can also use our AFM for the detection method. So nowadays, you know, there are a lot of big interest or big technology development to basically detect the different biomarkers. So what is a biomarker? Basically, it's the indicator of the disease states. It can be like a DNA, RNA, cell, bacteria, protein, enzyme, everything. For example, like now COVID-19 situation, the RNA is the biomarker basically to confirm or to indicate how we have this COVID-19 disease or not. So there are kinds of now many big interest in the field, how we detect those biomarkers. And one thing is to use this biomarker in the clinical diagnostic or preclinical research, we have to use the signal transducer. So like to detect it, we need the signal either to electrochemical, fluorescence, color, or the FET or mass temperature. Many things can be used for the to basically to get the signal in our sensor surface. And to get better resolution, we need, of course, we need a really better resolution to detect the lower amount of the molecule. We can also apply the AFM to do it so. So what would be the reliable quantification of those biomarkers in the biosensor is the first, we need the ultra-sensitive measurement. And also, the simple sample prep, and if we can handle really low amount of the sample, would be really ideal. And as we are kind of doing the biophysics, of course, we are also interested in the kind of kinetics or cooperativity in our binding or affinity thing. And I just brought the two common tools to measure those quantitative, reaction of our biological molecule. One is using the SPR, and then one is the QCM, quartz crystal microbalance. And when we see the both kinds of tool, then I just brought it in the catalog of the two company. What is the limit of detection and the limit of detection mass is basically we need a 1 femtogram or 1 nanogram of sample really to get the minimum signal change. And can we really improve it with the AFM? So we produce our DNA array sample on the surface. So here on the silicon or glass surface, we modify the 27 acid dendron. And this dendron structure is pretty good because it has kind of 10 nanometer spacing between the two apex of the dendron structure. So top of it and the apex of the dendron, we basically covalently link our capture DNA. as you can see here. And then we further basically put the target DNA with a different the concentration. So like from 10 picomolar to the 10 nanomolar, different concentration. And then we hybridize them. So for our study, we use the leukemia biomarker. And meantime, we also modified our AFM cantilever with the detection DNA here. So this detection DNA designed to detect the other end part of the target DNA. And this is the sequence. So detection is like we need a 20-mer DNA. And the target DNA can vary the different length, like but ideally 170 mers were kinds of ideal. And then capture DNA to capture we need again the shorter DNA on the surface. And to prove it really worked with the AFM, we first try with the force mapping method, which is the common method. So as I explained you before, this force distance curve, you remember it? And we perform this force distance curve, basically each pixel of AFM image. So one time up and down, and then go next pixel, and then up and down, and then next pixel, and so on. So each pixel means the one force distance curve. And total pixel is 128 by 128 pixel. It's about the 2 by 2 micrometer size. And when you measure this up and down in this 2 by 2 micrometer size, then it requires kind of for the one scan image, one map, we need kind of 70 minutes. And we collect the pixel where the rupture force shows between 20 to 40 piconewton. And basically, if there are more than one pixel next to each other, so like this, and then we measure the 3 times of this, and if they shows more than 2 times, and we start to count as a one cluster. And we increase our target DNA concentration, and then of course, as the concentration increased, our number of cluster also increased, as you can see. So we can really detect the one target DNA on the surface. The problem here is it takes some time, like a 70 minutes per one map. So to reduce the time and also to get the better resolution, we basically apply the another technique called the simultaneous topography and recognition imaging, so-called TREC, developed by our group. So this case is not this static movement anymore. So AFM cantilever is oscillating in their resonance frequency, in our case like a 10 kilohertz. So as it oscillates, you see the amplitude is like this. And when our molecule on the tip interact with the receptor on the surface, then that influence to basically this amplitude shape. So we can divide this amplitude upper part and then down part. So this downstroke basically get the information of the 3D structure of the surface. But this upper stroke is basically due to the stretching of interaction between the ligand and the surface. So here is the oops. Okay, the video is not working. Okay, video is not working. No. Okay. Sorry, the video is not working at this moment. So basically, this AFM cantilever scans like a normal AFM imaging mode, but when they interact with the molecule, what we are interested, and then this upstroke basically hindered by the linker or interaction or the bond formation between the ligand and then receptor. So therefore, this upper stroke goes down, or which is shown as a dark spot, as you can see here. And we apply this technique to our DNA sensor. So here's the AFM topography. You see we don't see where DNAs are, But when we get this recognition image simultaneously, then we could clearly see where our detection DNA ever interact with the target DNA on the surface. So here there are several dark spots. And when we make this background signal a little bit out, so we give the voltage threshold about the 0.5 volt when the peak-to-peak amplitude is about 1.5 volt. And we can see really clear dark spot. And then when we magnify it here, as you can see, the size of one spot is like a 10 nanometer, which is actually pretty good agreement of the Brownian motion of our target DNA when they link to the apex of the dendron. So that's about the 10 nanometer. And what is the main advantage of using track is as it's the same as we are using the just tapping mode, so normal imaging mode. The time of one map, one image is about the 4 minutes. So time-wise, we could really make it faster from the 70 minutes to the 4 minutes. And also we can increase our pixel number. So previously, each pixel cost the time in our case, which is not the case. So our case, the 1 pixel would become really 5 times or 10 times smaller than the previous conventional method. And when we measure the same area continuously, still most of recognition spots remain. But when we see the same spot after the 4 minutes later, next image, then the shape of this recognition spot change. But it's due to basically the probabilistic nature of our interaction when we scan the surface. So some pixel yes, some pixel not. But we could clearly reserve the single spot reserve after 4 minutes, even though after 4 minutes can happen, the thermal drift can happen and so on. So we also apply this track method to higher concentration target DNA hybridized surface, and then you see the recognition spot become bigger and also number is higher. And we also perform the blocking experiment. As I said, this interaction, the up-down stroke is due to the interaction between two molecules. So if we change the amplitude, it influences a lot to our recognition image. So when we have too low amplitude or too large amplitude, and then this recognition spot disappear. But also meantime, when we inject the blocking solution, so this blocking solution basically block the tip part, and then most of recognition spot disappear. So blocking worked well. And also when we washed it, and then most of recognition spot recover. So it shows the quite a good performance. And furthermore, we basically calculate the recognized area in one image according to the hybridized target DNA concentration. And then when we compare them between the number of clusters what we measure from the force mapping and the recognition area from the track image, we have quite a good correlation. And also from our recognition area map, the plot, we could calculate our equilibrium dissociate constant, which is about the 10 power 10 molar range. So finally, I want to bring it to the conclusion. So AFM is actually really good tool for imaging also the living bacteria cell on the physiological condition, which can really distinguish the single protein lattice structure. And also for spectroscopy or single cell for spectroscopy, we can basically unravel the really mechanism how this bacteria or cell interact with the host surface. It can be also abiotic surface with a different material. And then finally, our track basically shows the really powerful tool, the label-free quantification method. And in our case, we need a kind of 100 to 700 molecule to carry out the affinity, which compared to the SPR or QCM really require minimum number 10 power 4 or 1 million target DNA to get the signal. So our track AFM provides really the fast and label-free biosensing platform for the affinity study with especially the small sample amount. And we would like to apply this our technique also to other molecule or other DNA or RNA sensor surface. So here is the acknowledgement that they are my funding body and also collaborator with the different material. So the postdoc, they provide the dendron surface, and then we get this polymer brush sample and also different types of bacteria from 2 different universities. And of course, thank you for your attention.
Thank you very much, Eugene, for so nice presentation. So everyone is now welcome to ask the question. But I have also my personal question. Can I ask you, can we regulate, for example, binding of target, let's say my protein, which has active site somewhere deep inside of the protein? And can we regulate, for example, that this protein will bind covalently, not too close to the active site entrance, for example? Or is it stochastic process how we can bind this? Because otherwise, if I want to test some ligands binding, so of course it can happen that the entrance of to the active site will be blocked.
Yes. Yeah. So basically, we need a well-defined basically tagging part. So we can basically covalently bind the protein with a lysine residue or cysteine residue or histec part and so on. So you, this histec protein, you can modify where tag is, which is the other side of the binding pocket. Of course, if it's very close, and then it can have some difficulties. But when binding pocket exposed well, then yes, it's not the problem. So basically, what we could try is we could give the 2 different tag, so one 3 end, one 5 end, and then we can see the different whether where binding pocket is. But yes, it's actually good to know before we measure where really binding pocket is and where we basically link it to our AFM cantilever. Was it the question? Yeah.
Yes. Thank you very much. I have also another question. Is this protein during these measurements, when it is already linked, is it in buffer solution?
Yes. Basically, everything is in the buffer. Today what I show you, everything measured in basically buffer. It can also vary. So you can use normal PBS or TBS buffer, but we can also try other buffer. Just the thing is as we have to detect the movement of the cantilever through the laser, if it's really not transparent buffer, so for example, like a real blood, then we may not see well. Yeah. So if it's a transparent buffer, then it is possible.
And for example, if I have some microcrystals of the protein, is it possible to work with them?
Microcrystal. I mean, it doesn't have to be protein or, basically we can also bind the other small size if there is a tag, like it can be nanoparticle, of course it's possible. And also polymer coated, yes, it doesn't have to be, the protein. Or, I mean, even DNA is not the protein, right? So,
yeah.
So
and
it works.
And just to be able to produce this experiment, what the protein, what is the percentage of the binded protein should be rather than non-bind? Because of course it's always ratio. You cannot control this process.
I mean, to prepare this AFM tip, covalently binds, we have like 90% are working. Yeah, if really the tag is clear, then it's the very clear chemistry step, so it work quite well. But of course, it's depending on the. Then next issue is the material. Of course, it's a statistical experiment, so we do many times. But yeah, if we basically have to know, I mean, this binding probability is other issue, because then it's protein interaction or whatever interaction happen or not. That's I cannot give the number. But our chemistry step to link the molecule, then we have kind of 80-90% success rate. So, and we also kind of need rather kind of pure sample. So purity is also the issue. Yeah, it should be kind of more than 80% purity, of course.
And can you check for somehow that my sample is bind to the surface?
Oh yeah, then if it tag well to the cantilever, yeah, then we can just perform this
just run the experiment.
Yes,
yeah.
yes,
and
then see the whether, so like this how bacteria binds or not with whatever polymer surfaces. That's very clear step. You basically put bacteria and then do it, and you know it in one day.
Okay.
Bigger one is easier than smaller one.
I see. Okay, who else wants to ask the question? It looks like everything was clear for everyone. It was really nice talk. Thank you very much.
Thank you.
So the last chance to ask the question. Okay, so Mark, please. Yeah, try to ask.
It was very interesting.
Pardon?
Can you really a bit raise your voice?
It was very interesting.
Thank you.
Microphone is on.
Yeah, unfortunately, we cannot hear you. Try to switch off and switch on the microphone. Yeah. Try to speak right now.
I'm trying to speak now.
Yeah,
can you hear me?
Yes, we can hear you, but not perfectly.
Yeah, it was very interesting. It's I'm a little bit on the fringe of your research, but it was very good, understandable, and it was new to me as a recent usage and improvement of atomic force microscopy and other biological research. Thank you.
Thank you.
Okay, who else want to ask the question? Okay, if no one else wants to ask, so it looks like that everything was crystal clear for everyone. I would like to thank you one more time for so nice presentation, very informative, very understandable, and really on very basic level for everyone. So thank you one more time.
Thank you.
So Pavel, do we need to close somehow this session?
Yeah, I would like to thank to both presenters. I think that both lectures were really nice. In my personal case, I was quiet since yeah, I know AFM, of course, and stuff like that, but I'm not coming from the field, from the biological field, so I was quiet from that reason. But I would like to thank to all of you also for the participation. I think that we had really nice attendance, almost 50 people attended at some point. And I would like to invite you also to the second round of NABIAM lectures, scientific lectures. As I said, it will be organized at the end of January. We are now negotiating with speakers, and we will tell you in enough advance where when it will be take place. Thank you for your participation, and thank you, Tatiana, for moderation of this session.
Yeah, thank you very much also for very nice talks and opportunities. And we are looking forward to hear one more somewhere in the end of the January, some more lectures. Yeah, thank you very much.
Thank you.
And hope to see you everyone very soon.